Welcome to the Roots Lab. In this lab, we're going to shift our focus from what's happening above ground to what's happening below ground, inside one of the most important and often most overlooked plant organs, the root. So far in this course, you've explored plant cells and tissues, and you've seen how those tissues are organized in stems and leaves for support, transport, and photosynthesis. Now we're going to look at how those same tissue systems are organized in roots to solve a very different set of problems, like anchoring the plant, absorbing water and minerals, and interacting with the soil environment. Just like the Leaves Lab, this activity is designed to mirror what you would do in a face-to-face lab, observing prepared slides, making labeled drawings, and connecting microscope structure to whole plant function. By the end of this lab, you should feel confident identifying the major external and internal features of roots, comparing monocot and eudicot roots, and explaining how roots are adapted for transport, growth, and symbiotic relationships. Before we begin, let's take a moment to review the learning goals for this lab. By the end of this activity, you should be able to identify the major parts of a root and the key tissues you'll see in cross-section. You'll describe and compare different types of root systems like taproot and fibrous systems. You'll also focus on two especially important tissues, the endodermis and the pericycle, and explain not just what they are, but why their locations inside the root are so critical to how roots function. Finally, you'll look at examples of specialized roots and explore some really important root symbioses, including nitrogen-fixing nodules and mycorrhizae. Just like in the Leaves Lab, the big theme here is that structure supports function. Every tissue you see is where it is for a reason. So why spend an entire lab on roots? Roots do a lot more than just hold the plant in the ground. They anchor the plant, absorb water and minerals, and serve as the entry point for nearly everything the plant needs from the soil. They also play a role in transport by feeding those resources into the vascular system where they can be distributed throughout the plant. Roots also produce hormones that help regulate growth and development, especially the balance between root growth and shoot growth. And on top of that, roots are the main point of contact between plants and a huge community of soil organisms, some harmful, but many incredibly beneficial. Even though you rarely see them, roots are constantly making decisions that affect the entire plant's survival. Unlike leaves, roots don't contain chlorophyll, which means they can't carry out photosynthesis. Instead, they depend entirely on sugars produced in the leaves and other photosynthetic tissues. Those sugars are transported down to the roots through the phloem. At the same time, the roots are responsible for absorbing water and minerals from the soil and sending them upward through the xylem to the rest of the plant. So there's a constant two-way exchange happening. Shoots supply the roots with energy, and roots supply the shoots with water and nutrients. This makes roots and shoots completely interdependent. Neither can function properly without the other. As we go through this lab, you'll see how the internal structure of roots supports this constant flow of materials and keeps the whole plant system running. Over the course of this lab, you'll move from the outside of the root to the inside and from whole structures down to individual tissues. You'll start by comparing different types of root systems like taproots and fibrous roots. Then you'll examine the regions of growth at the root tip and see how roots lengthen and differentiate. You'll compare the internal structure of monocots and eudicots, observe how lateral roots form, and explore some of the most important root symbioses, including nodules and mycorrhizae. Throughout the lab, you'll be making careful observations, creating lab drawings, and connecting what you see under the microscope to how roots function in the real world.